we are going to have a presentation from someone who has been a part of our organization on the periphery for a while. Another person who we've met probably about 2 years ago now. Her name is Yael Weiss, and Yael has been referred to. I think that one of the best explanations that I've heard of was Eric and I attended the Rare Entrepreneur Boot Camp that Yael puts on, which is this incredible gathering of patient advocacy organizations, industry folks, capital, just this incredible collection. And I believe someone at the conference that we came to referred to Yael as kind of the fairy godmother of the rare disease community. And because she's not only incredibly knowledgeable, but she's incredibly kind in giving of her time to really educate in a kind way, and when needed to be very direct with me. I know that she's given some very blunt advice in the past, which is appreciated because we don't have time to waste. So, Yael, thank you for being here and presenting today. I'll turn it over to you.
Thank you, Jeff, for your very kind introduction, overkind, I would say. And thanks, Eric, as well. And thank you for having me. And I want to make sure that you're seeing the right screen. Can you see my presentation mode and not my emails?
We can.
Okay, great. So, hello, everyone. And I'm actually glad that my presentation came after Scott's and Melissa's, because I think what I'm going to talk about actually takes their information, the information that they shared with you, one step forward. And I'll talk a little bit about drug development in general and not go specifically into Kleefstra details, but I'd like to talk a little bit about how to form a collaboration between patient groups, academics, and biotechs, and how we try to do this through the company that we founded, Mazi Therapeutics. So let's see if I can advance my slides here. Yep, I can. So I'll give one slide about Mazi just so that you understand where we are coming from. And Mazi was founded based on interactions with people like Jeff and Eric that we met at boot camp, and saw the dedication and the strong motivation, obviously, to develop therapies, but not always having the full set of capabilities, expertise, and funding to take any of the programs that were funded forward. So Mazi was started in order to work with advocacy groups to look at what they have funded in the past or help them start new projects that could potentially be translatable, taken into the clinic, and become products. And we are a small team, all experts in rare disease, all been around for a long time, only 12 people at the moment. And the way that we operate is that we work with advocacy groups, and you can see on the top left the 3 advocacy groups that we're working with now that have the most advanced programs. We do have 7 or 8 additional groups that we work with regularly, including working with Jeff and Eric on Kleefstra. We work with the Walks Foundation, the Pitt Hopkins Foundation, and the Coalition to Cure CHD2. We work with the academics that actually are doing the work that is funded by the advocacy groups. And if the work is successful, and I'll talk a little bit about what successful means, then we would bring it into Mazi and continue developing it. So we have collaborators from the Hebrew University in Israel, UC San Diego, and the Weizmann Institute in Israel. We work with diagnostics companies. I haven't been present on all the calls, but I'm sure if it hasn't come up yet, there will be a conversation on finding more patients so that we understand the disorders better. There's a better understanding of the epidemiology, better understanding of the clinical presentation, and also a larger pool of patients to enroll in clinical trials and to sell the drugs to eventually. So we put a lot of emphasis on projects that will enable us to diagnose additional patients. And we work with CROs and CDMOs to execute the programs. So this is the only slide I'll show about Mazi, and I'll jump into the rest of my talk. So you've heard a little bit, and you'll hear more about how drug development is actually not for the faint of heart. It's complicated. It takes a long time. It takes a lot of funding, and many times we fail. So we have to just go into every project that we start knowing that there might be a failure in the end. So we need many shots on goal. We need to have a good understanding of the disorders that we're planning to treat. And drug development in rare diseases, especially in rare diseases that have never had a dedicated specific drug for them, are more complicated because we have to understand the different endpoints in a clinical trial. I'll talk about that a little bit. We have to understand the natural history of a disorder. So, and if we're the first company that's taking a therapy for a specific disorder into the clinic, we have to make sure that we work closely with the advocacy group to hear what you as parents, as families, would like to see change in a clinical trial versus what we assume is going to change. So the bar is even higher than a drug for heart disease or asthma, obviously. So proof of concept, which is what usually we see coming from advocacy groups, is only the beginning. There's a long way to go after that. And the partnerships between all the parties in this development path, I mean, it's a friendship forever. It's almost like a marriage. Once we commit to develop a therapy for a specific disorder, we're with you in it. And it's critical that it's a good relationship, a productive relationship, and each partner understands what they bring to the relationship. There are many tracks that go in parallel, and I'll show a slide on that later. The research that is funded, a lot of research is basic understanding of the disorder, but at some point, some research needs to be done that's translational and could switch into a drug versus understanding the biology of the gene and the protein. The goals between the different parties need to be aligned, as I said, and we all need to have the same sense of urgency that you have. And if we don't have it and we're not aligned with you, then you'll be very frustrated with us. But to the scientists on the call, I say, this is why we became scientists, right? We wanted to make a difference in people's lives. So we need to keep that in mind. The topics that I'll cover are, what is a target product profile? So when we start developing a therapy, we need to know what the drug will look like at the end. And I'll show you what the parameters that we think about are so that you understand why we ask you all these questions and why we do surveys and why we do interviews, so that we understand what this drug will look like at the end. So this is our the sign at the end of the way that we see, and we have to gear all our development towards it. What is the science? What is the modality? How do we choose which modality to use? So you heard a lot about ASOs. You'll probably hear about other modalities in the rest of the days. What is the scientific data that needs in order to fulfill this target product profile? And what is proof of concept? What do we do after we have the cell and animal proof of concept? As I said, there's a long path. I'll talk a little bit about that and how we work together with all our partners. So this is the target product profile. So what you see is in the center, there is the target that we're hoping to get to. We do set a set of minimum criteria in case we're not able to meet the target, and we do set an aspirational criteria that would be the best case scenario. If we can get to that, that will probably be a much better drug than the target drug, but we can't always make it there. So the first thing that we need to populate in this table is how the drug works. Is it an antisense oligonucleotide that upregulates the mRNA, just like Melissa just described to you? Is it an AAV gene therapy? Is it a small molecule? We need to obviously describe what the modality is and how it works. What does it do? What is the indication? Are we saying we're going to treat all symptoms of cleftra, or are we going to treat only part of them? We have to define that from the beginning based on what we know about how our drug is working. Who is going to participate in the study? Are we going to enroll everyone, or are we going to enroll only kids or adults with specific symptoms? What are the ages that we're going to enroll? We have to define this up front because this is how we'll define the output of these studies as well. Is the drug working? So what is our primary endpoint? What is the first thing that we're looking to see a change in? And what are other endpoints that we're hoping to see a change in? And then are there any exploratory endpoints that we think maybe we'll see a change in, but we're not sure? What is the pharmacodynamics of the drug? How does it distribute in the body, and how long does it stay in the body? And then, of course, is it safe? Could it be given with other drugs, or are there any contraindications? Are there any warnings that, if somebody takes the drug, we should be paying attention to specific symptoms that might occur post taking the drug? And are we anticipating any serious adverse events? And of course, how is the drug supplied, storage, handling? So we have to think of everything in the beginning, because if the drug can only be stored in minus 70 degrees and a family needs to administer it to the child at home, do families have minus 70 degrees freezers? And so this is just information that we need to set. And then when we think of how the drug will be distributed or given, make sure that whoever does it has all the right storage conditions. So what are the different therapeutic modalities? I don't know if this was covered. I wasn't on all the talks previously, but I will go through it quickly. The three big buckets of therapeutic modalities that are available now are small molecules, which is like aspirin or NyQuil or any drug that you take and put in your mouth and swallow. That's usually a small molecule, a chemical. There's a gene therapy, which is at the moment a one-time therapy that's injected into the CNS. A viral vector is integrated into the cells, into the neurons in the brain, and makes protein. So that's a replacement of the missing protein or the mutated protein. And the antisense oligos, which you heard a lot about, and I won't go into too much detail about them. So just looking at this very quickly, and I'm happy to share the slides afterwards if somebody would like to go into this more deeply. When you look at the mutations treated, small molecules don't usually treat the mutation specifically, maybe indirectly. Gene therapy usually treats loss of function. So when there's protein missing or mutated and not functioning perfectly, then we will be bringing in more normal protein. And antisense, as Melissa said, can work for knockdown, so loss of function or gain of function. Small molecules usually given in the mouth. That's the most popular way as a pill, but also could be injected. Gene therapy is a one-time therapy. As of now, there may be in the future developments that will allow re-administration, but at the moment, because the body immediately responds, there's a strong immune response, we could not give it more than once. And antisense oligos are usually given 4 times a year or sometimes every few months, depending on the half-life of the molecule. How do these drugs distribute in the CNS? Small molecule needs to cross this barrier that is between our blood and our brain. And if it does cross, then the distribution is very good. Gene therapy is, even if you give it straight to the brain, it usually circulates in our brain and goes back to the rest of the body through the blood. So the distribution can be limited, and antisense oligos distribute very well. What are the known limitations of each modality? The small molecules, as you know, they're anti-seizure molecules, they're attention deficit disorder molecules. There are many small molecules, but they don't really change the course of the disease. They treat specific symptoms. We don't usually know, especially if it's a repurposed drug, we don't know how it works in a specific disorder. We need to interrogate that. And there might be some off-target effects, meaning that it binds to what we want it to bind to, but also to other things, and then it gives effects that we're not necessarily happy with. Gene therapy has its limitations. I won't go into too much detail on that. If we want to have a seminar on gene therapy, I'm happy to jump on and do that separately. And antisense you heard about, it has some known adverse events, but it's also a very efficient therapy when it works. The cost, and this is, you heard about probably gene therapy for SMA. This is a big discussion because the cost of some drugs is very high and it may prohibit access. And this is one thing that we would like to avoid. While pills are very cheap and antisense oligonucleotides are somewhere in the middle, the gene therapy modality is very expensive. It costs millions of dollars, taking into consideration that it's given once in a lifetime. If you divide the cost over a lifetime, maybe it's not so expensive, but there's definitely a sticker shock here when you see the price and you hear about it. So again, another table that helps us decide which modality to choose. I won't go again into too much detail, but I'd like to talk a little bit about why gene therapy isn't always the most suitable modality and why an ASO isn't always the most suitable modality. Gene therapy has practical limitations. Sometimes the gene is too big. It won't fit in the delivery system that we put into the brain. Sometimes there are many different transcripts, many different isoforms of the gene, and we don't know which one to choose. Do we need to know where the gene is expressed, how it's regulated? Sometimes we do, and sometimes we don't. So these are the parameters that we look at when we look at the gene before it was mutated. Then when we look at mutations, we need to understand how the gene is inherited. Is it a familial mutation that is passed from parents to the child, or is it a new mutation? And then maybe it hasn't even been described before, and we need to understand what the consequences of this mutation are. Is it a loss of function, gain of function? Is it a missense mutation or a nonsense mutation? Will it lead to no protein at all, or will there be some residual protein activity? That's really important to know because we need to know how much we're aiming to augment. And then the genotype-phenotype correlation actually talks about where the mutation is in the gene and how it presents in the clinic. So a child might have no protein at all and have a certain set of symptoms, and then a child that has a little bit of residual protein may present differently. And then there may be situations that we can't even explain by looking at the correlation between where the mutation is and what the effects of the mutation are and the clinical presentation. But this is something that we try to understand before we go into a clinical trial. And then what the protein function is. In some cases, it's great to know what the protein function is, but it's not always mandatory. So I'm moving on to proof of concept because many scientists who come to us and say, oh, we want to work on this syndrome, what do you need to see in order to make it attractive for you to develop it as a drug? So it's not really a one size fits all. It depends on the gene, it depends on the protein, it depends on the disorder. But we would like to see data from human cells, and I saw some of this data presented this morning. And ideally, if there is a good mouse model that represents the syndrome, to also see data in a mouse model. Cells could be either isogenic cells or patient cells. And if there are organoids, so 3D structures, that's really helpful. And the mouse model, in many cases, it's not representative of the disorder. Mice are mice, humans are humans. It's hard to expect a mouse that is mutated to behave just like a human that's mutated, but some symptoms might be reproduced in mice. And if we can show with a drug that we eliminate these symptoms, then we're in a good place. And then if there is a biomarker, something that could be measured to predict the activity of the drug, that's really great. This is a quick example of what we received from an investigator and made us choose one of our disorders, worry syndrome. You can see, if I can find, see if I can find the laser pointer. So the middle mouse here is the mouse that has the mutation. It's very small, it doesn't develop correctly, and it dies at a very early age. It seizes and has a lot of the characteristics that are similar to those of Walk's-Worry patients. This is a normal mouse that has no mutation, and this is the mouse that received the therapy. So the scientist was able to show us that by giving the mutated mouse a gene therapy, he was able to restore the lifespan of the animals. They grew and developed as expected. They stopped seizing, so they normalized. And this gave us a lot of, gave us a lot of promise that this drug hopefully will be effective in humans as well. So this was a pretty comprehensive data package that really convinced us that this might be a good drug. Again, moving from mice to humans, not guaranteed, but this is as good as we could get for this specific disorder. You heard from Scott about Angelman, so I'm not going to go into that too long, but just focus on the fact that in this case, FAST started a company and the company was acquired. It's not necessarily a model that everyone needs to adopt. Some people could come to a company like Mozzie. There are many different models where research could be advanced. In the case of Angelman, this was the model that they adopted. And I just wanted to show, as a follow-up to Scott's presentation about the ASO and how he discovered it, this is what the clinical trial looks like now, and how the patients are dosed, and I'll show you what the endpoints are. But in the beginning, the drug is dosed more frequently until a stable dose is achieved, and then dosed every 6 months. So in every clinical trial, and even in reality after a clinical trial, there will be a loading period where the drug is loaded until it reaches a maximum dose that is tolerated, and then the frequency will go down. And this is an interim data that was presented by Ultragenyx, and I just wanted to use this to show you what things were measured. So there are a lot of domains that are being measured: behavior, sleep. I'll go back one. You can see sleep, behavior, communication, gross motor skills, fine motor skills, and overall. And all these traits are being consolidated, and each patient is observed to see what are the best or the most significant changes that were achieved by using the drug. So in disorders that are new to the clinic, it's not clear what the endpoint might be. There is an ability to consolidate many different issues that are bothering you as families, put them on a table, and see which ones move once the therapy is advanced. After clinical trials, we have to go through a whole set of additional experiments to show that the drug is safe, to show that it distributes properly, and it's usually done in mice and in monkeys. It takes about a year to a year and a half, depending on what the output of these studies are. So this is why it takes a long time to get from proof of concept into a clinical study, because there's so much more that we need to do in the middle. And this is just a slide that shows how we interact with the regulatory agencies. We start talking to them very early, even when we have very little data, but just enough to suggest what the target product profile that I showed you in the beginning looks like. And then we take these, after we interact with the regulatory agencies and get an idea and get feedback from them of whether they approve our approach, we start the IND enabling studies to show safety, to show that the drug distributes, as I said, and to have a confidence that the drug is not only working, but it's also safe to administer to humans. Clinical development, very quickly. As I said before, we're assuming that there's been limited clinical exposure before, and that the diseases that the therapies that we're working on are disease modifying. So what are our challenges? Our challenges are validating new endpoints, or taking an endpoint that was validated for another disorder and making sure that it actually is relevant for the new disorder, because seizures may be different, communication might be different, motor skills might be different. So a measure that was built for one disorder has to be adjusted for the new disorder. So we take a lot of time to understand that. And then the studies are small because the disorders are small. There are not a lot of patients. So the small studies, they're disappointing to the community because we don't enroll a lot of patients. But the reason that we conduct small studies is that we can enroll them quickly, see an indication of efficacy, and move as quickly as we can to dose more patients in the next trials, and then obviously in the commercial land where we are actually selling the drug. And when we start putting together the trial, we collate input that we received from patients, patient advocates, families, from the treating clinicians, and from the regulators. So these have to align into a cohesive clinical protocol. And as I said before, we hope to have CSF and blood as well, and some biomarker to show that very quickly can give us an indication whether the drug is working or not. This is what we go through when we work with advocacy groups. We start by giving a caregiver survey to understand what a day in the life of a family looks like. We look at medical record data, and we review those to try and get an assessment of what happened a few years back. Then we interview patients, long interviews for an hour or so, and really go through all the information that we collected from the first layer. And then we plan a natural history study, which would be moving forward. We would put a study in place where we would enroll patients and collect information instead of back information, new information moving forward. And this usually takes about a year or two. And we started early in the process of drug development for a specific disorder. And then we have a model that really gives us all information about the disorder and helps us design the clinical endpoints. So patient groups, this is something that I share with patient groups when I meet with them, what the advocacy groups can do while we're developing the drug. So advocacy groups contribute by, first of all, being a cohesive community, and that anytime that Jeff and Eric send an email and request something, that it gets to the majority of the known patients so that we can collect as much information as possible. The patient groups usually have a registry. In some cases, they have a natural history study, not always. And if there isn't one, then we will start one. They will participate. Obviously, you will all participate in the surveys, and you will help us enroll in clinical studies. You will help us define what meaningful clinical endpoints are. You'll help us find biomarkers, because you and your kids will give us blood samples or saliva or whatever is needed to look for a biomarker, and you will help us design the disease concept model. You will be donating cells that will be made into organoids and possibly banked, and you, by sharing your mutations, will help us design animal models that carry these mutations and are relevant for our studies. And then we will take on everything on the right-hand side and do everything that I talked about up till now. And this is just the parallel tracking. We don't do things sequentially. We try to do everything at the same time, so that when we want to start a clinical trial, everything is ready. Clinical endpoints, knowing where we want to place the clinical studies. Is it where there are patients? Is it where the clinicians that see a lot of patients? Kick off the registry right off the bat, start finding more patients right off the bat, and then prepare all the research tools that allow the proof of concept right in the beginning. And you're definitely on the right path for that. So the goals need to be aligned between academics. Academics in many cases want to publish, they need grants, they don't have the sense of urgency that advocacy groups have, while families have all the knowledge and information and the sense of urgency, but not the abilities to do what the academics are doing. So there needs to be alignment, and very clear alignment from the beginning. So when a group like like I defined funds research, they need to put timelines in place, expectations in place, and make sure that the scientists that you're working with actually perform and understand what the goal of the research is. This is more or less what I said already, so I'll skip it because I see I'm running out of time. And in summary, it takes a village, and really, literally a village, to develop a therapy. Alignment means collaboration and synergy, and the keys to success are solid biology from the beginning, which you have, smooth handovers between the advocacy group to the scientist to any biotech that takes over the project, no gaps, but very collaborative, parallel tracking, as I showed in the slide, and a lot of collaboration. And thank you. And if you have any questions, if you want access to the slides, please ask questions or tell Eric and Jeff that you want my slides.
Thank you, Yael. Really fantastic. I think it's encouraging for me, I think, just seeing a lot of the things that you're saying that we need to be doing, that we're actively, I mean, partially because of your encouragement and your guidance and direction, if not for all of it. But a lot of those things that you listed that we need to be doing, from the data collection to the biomarkers to disease concept model, like those are all things that are being discussed at this conference over the next couple of days. And so we just need the responsiveness. We need the community to lean in, and I think we're building this ecosystem with folks like YaEl, with folks like you've heard today and the next coming days, that we're assembling the team, we're developing the roadmap, and we're putting the resources in place where we're putting ourselves in a position for these things to happen. Right? There's no guarantees, but we're doing the work, and it's again, there's like a validation when I see that we're on the right path, and we just have to continue pushing forward. So amazing. Yeah, I think you're fantastic, and I can't thank you enough for everything you've done for us.
Well, thank you for having me. You're definitely on the right path. And if anyone has any questions or concerns, please send them my way.